too much. Now we have another speaker, Mr. Vijay Singh, Dr. Vijay Singh from University of Queensland, Australia, and he would be talking on, she would be talking on genetic control of nodal root angle is associated with drought tolerance in sorghum. So the floor is yours, Dr. Vijay Singh.
They said hello. It's on. Okay. Okay. First, I'd like to thank the organizing committee for giving me this opportunity, and I'd also like to acknowledge my co-authors, especially Graham Hammer and Eric, who are sitting in this audience. So my key points from this talk is that seedling nodal root angle is an indicator of root system architecture in mature plants in sorghum. Nodal root angle influences water extraction pattern and is under strong genetic control. Breeding populations have been screened using high throughput systems and controlling genetic regions identified. There is significant association of these regions with yield in dry land breeding trials. I will not go into the detail about the methodology, but these photographs shows root development of sorghum. Yeah, this photograph shows root development of sorghum. Unlike other cereals, sorghum produces only one seminal root, and the first pair of nodal roots start to appear around 5-leaf stage. This was considered critical for root screening, and genetic variation for nodal root angle was observed. So knowing the differences in nodal root angle at seedling stage, we designed a large rhizotron to link these differences at seedling stage with root distribution and water extraction pattern of mature plants. These big chambers can hold around 400 kg of soil. So I've just put together a few photographs to show how intact 400 kg soil was transferred on the pinboard. Selected lines differing in only nodal root angle but had similar phenology and tillering were used for this experiment. This photograph shows contrasting root development pattern between the two lines. So if you see on your left, the narrow angle line has more uniform branching, better occupancy, in contrast to wide angle line, which has very shallow root system. Again, the next experiment was conducted in the same large rhizotrons, and gravimetric water content was measured at anthesis from 20 by 20 centimeter grid. The whole chamber was divided into 3 sections, so 0 to 80. So the x-axis is distance from the plant line and y-axis is the percent of total water extracted. 0 to 80 is just below the plant line, and 160 to 240 is furthest away from the plant line. And the light blue bar and the red bar represents the narrow angle line, and the black bar represents the wide angle line. If you see here in the figure, the narrow angle line tended to extract a lot more water from just below the plant line, that is the 0 to 80 centimeter. In contrast, the wide angle line tended to extract a lot more water from furthest away from the plant line, that is 160 to 240 centimeter, showing that there is difference in water capture between the two lines. So knowing the importance of nodal root angle at seedling stage, its influence on water extraction pattern, we designed this new phenotyping platform. This phenotyping platform has small, again, rhizotrons. We hold these chambers in large steel tubs. Each of these tubs can hold around 50 of these chambers. We have 10 of these tubs, so we can screen around 500 lines in one run. We also have an imaging box, and we can image all these 500 lines in a day. Initially, we screened around 70 inbred lines, and you can see here in this photograph, there is a big variation in nodal root angle between these lines. This varies from 15 to 55. Then we screened around 213 drill population, and we identified 4 nodal root angle QTL colored by green. These QTL explain around 58% of the phenotypic variation. There was no genetic linkage between the nodal root angle QTL with plant size QTL. And however, these nodal root angle QTL co-located with stray green QTLs. This table shows that QTL for nodal root angle associated with yield data in breeding trials. So there was a significant association between these markers with yield in 3 different hybrid combinations. The genetic regions controlling nodal root angles are having an effect on yield, and yield was increased in the presence of narrow root angle QTL. This figure shows the simulation results. So there is higher relative yield increase in narrow angle line compared to the standard variety. So if you look here, the narrow angle line had higher yield, especially under lower yielding environments. So recently we have screened around 3,000 lines from 3 complementary population developed by sorghum breeding program in Australia. And this box plot shows the variation in nodal root angle in the 3 population. And you can see here, the diversity panel has the widest variation, and this narrows down as we move from diversity panel to more elite lines in the breeding population. The heritability is very high. The higher heritability for diversity panel and NAM population explains greater genetic variance observed. We also have high density SNP data for all of these 3 populations, and we have combined this with phenotypic data to identify regions of genome significant for nodal root angle between and within the populations. We have identified 30 different regions between the population and 5 regions in common between all the 3 populations. So now currently we are mining these regions for candidate genes. For example, we have found RO1 on the NAM population, which is in common with QTL for root angle in maize and rice. So in summary, I've shown you that seedling nodal root angle is a good indicator of root system architecture in mature plants. And we have found that nodal root angle influences water extraction pattern and is under strong genetic control. We have screened already 3 breeding population using the new system. So it looks like the new high throughput system is working. And we have shown that there is significant association of this region with grain yield data from different breeding experiments. And finally, I would like to acknowledge all my colleagues, because for transferring 400 kg soil, I don't think so I'm big enough to transfer it, and all the funding bodies who have supported this work. Thank you.